Thank you, Alex. Good morning, good afternoon, good evening, depending on your time zone. Today, we're going to discuss some points to consider when determining the level of detail needed for the CMC portion of your application for a small molecule, biologic, and vaccine. Great. There we go. So we're going to have a brief introduction to the topic. We're going to look at some considerations for the small molecule. We're going to discuss the appropriate regulatory detail for a biologic, and then we're going to go and discuss some considerations for a vaccine, IND, and BLA application. And then we'll have some time for some questions. We don't need to go through that. We already discussed that. So you need to make sure that you include the right level of detail in your NDA or BLA, because too much detail, I've heard that if you don't receive at least a few FDA questions, then you've probably included too much detail in your application. And adding too much detail can also cause the FDA to ask additional questions. They have more detail to ask questions about. Too much detail will make post-approval changes a lot more difficult. So when considering the level of detail, make sure that you add details specified in the FDA Regulatory Authority meeting minutes, but don't go overboard. My colleagues in the biologic and vaccine application realm will comment on this also. You want to make sure that you review ICH M4Q and all the associated ICH quality guidance documents. But remember, and this is from 314.70A1I, the applicant must notify FDA about each change in each condition established in an approved application beyond the variations already provided for in the application. And we'll discuss that a little bit more later too. During this presentation, we'll look at some considerations to make when completing an NDA or BLA for a small molecule, biologic, and vaccine. I already said that. Variations on the level of detail discussed will be dependent on your actual application. Key points to always include, no matter which type of application you're going to file, are items pointed out by or discussed with the FDA in your pre-NDA, pre-IND, and your CMC meetings with the regulatory agency. It's a good practice to have both a senior level CMC person attending these meetings, as well as a not so senior member who could just take copious notes and of course learn. These notes, along with the FDA written minutes, will help you identify exactly what level of detail the FDA is looking for in your application and which parts of your application they may be most concerned with. The next item we need to consider is to look at what modules are regulatorily binding. That is, which modules will require a post-approval submission to the FDA if changes are made to them. Understanding this concept is important, as you want to make sure you don't adversely impact your ability to make post-approval changes to your product by putting in too much detail. You also want to know your FDA project manager. Keep notes on any snippets of their home life that they might share with you, so you can start a conversation asking about these points in the future. I read a science fiction book by Robert Heinlein who talked about a Farley file, which included just this. He would keep notes with the person's name and just the snippets of what was going on in their life. Ask if they know who will be reviewing your application, which is not likely, and if they have any insights about the types of items this person or persons look for. People that have a history in drug packaging will tend to be more involved in the drug packaging than in other sections. Another item to consider is comparability protocols. These will be discussed by my colleague who will be discussing the vaccines. So selecting the right level of detail for your application is a moving target. It's hugely dependent upon the complexity of your drug substance, drug product, and any dosing device or devices. The higher the complexity, the more detail is needed to demonstrate identity, strength, quality, purity, and potency. Knowing which modules are regulatorily binding and which are not is very important when considering the level of detail to include in each section. Modules that are not regulatorily binding are usually modules that will benefit by including additional detail. For example, the more characterization information you can include for your drug substance, the more data you have to fall back on when you need to make a change to your synthesis process. you have something, you have data that shows you can compare to, and avoid possibly having to do a clinical bioequivalence study. Adding extra detail to establish conditions sections may help with significant changes down the road. You will have more information, again, as we just spoke about, to compare the results pre and post change. Modules that are regulatorily binding are the ones you really need to ask, is this the right amount of information? Is there enough information provided for the FDA to make the determination that the product is safe and effective for human health? Is there enough information to confirm identity, strength, quality, purity, and potency? Also, for manufacturing modules, you do not need to include detail that's GMP. GMP information is a given. You only need to include detail that is specific for your product. So this is a new slide that you don't have in your slide deck. This is from ICH Q12, figure 1, and it shows a little bit about what you need to do, the decision trees that you make when you have a post-approval change. And so you're going to first ask, is it an established condition in my filing, or is it not? And if it's not an established condition, then you don't need to update it. If it is, then you do need to update it, and you have to make the decision about whether it's going to be prior approval or just require notification. And this is another similar slide. It's also from ICH Q12, and it talks about what the kind of work that you need to do inside within your company, and then if it ends up being an established condition, what the regulatory process is for that. And again, this is all in ICH Q12. It's a good document to be familiar with. So now that we have a little bit of information on the established conditions, we're going to go through a couple tables that will discuss which are established conditions and which are not established conditions. Okay, so nomenclature, structure, manufacturers, description of the manufacturing process and controls, control of materials, controls of critical steps and intermediates. These are all regulatorily binding modules and will all require an additional thought. You know, is there too much detail? Did I include too much information here? Process validation is not regulatorily binding, as every time you make a significant change to your process, which would require a post-approval submission, you should revalidate. The original validation will serve as a comparator to ensure you're hitting the same critical quality attributes. Any CQAs that are no longer needed as a result of the manufacturing change should be noted in 32S24 and will also require prior approval. The process validation is backwards looking. So another way to look at whether a module is regulatorily binding or not is to look, does it look back? Does it say what was already done, or does it say this is what we're going to do in the future? 32S31 and 32 are also not regulatorily binding, as they consist of baseline data that you would perform again when you're making a significant change to your product or process. If your drug substance or product does not test as equivalent using the characterization and impurity methods listed in your application, you may need to perform additional clinical bioequivalence studies to demonstrate equivalence. And again, 32S43, 44, and 45 are all backwards looking modules. Established conditions are regulatorily or legally binding information considered necessary to assure product quality. Any change to an established condition necessitates a submission to the regulatory authority. And that the last two sentences that I just read are both from, again, ICH Q12. So of note, section 32S72 and 32P82 post-approval stability protocols are not considered established conditions. It's more of a regulatory commitment. This is the protocol we will follow for post-approval stability testing. And then the 32P documents that are regulatorily binding, as you note, what's regulatorily binding is in red, and the established conditions are in the black. Okay. So 32A1, facilities and equipment, is usually submitted as used to be what was known as a site master file. It's regulatorily binding. It is also a nice to have for small molecules, but for large molecules and vaccines, it is definitely regulatorily binding, and it's definitely required in more detail than you would require in a small molecule. And that will be discussed by my colleagues also. So let's talk about a few items for oral solids. Okay, so criticality of an item. This is the first and foremost key question in ascertaining the level of detail required for your application. You need to include sufficient detail to convince the regulatory agency that you're able to consistently manufacture your product to the same level of identity, strength, quality, purity, and potency, whether it's summer or winter, the humidity is hot, it's cold outside, whatever. For tried and true synthesis or manufacturing processes, you don't need to include as much detail as you'll need to include for novel synthesis or manufacturing processes. The more familiar the FDA or the regulatory authority is with your particular synthesis route, your manufacturing process, the less detail they really need, because they understand the process a lot better. For example, for a drug product manufacturing tank, you should list the material of construction and the validated size of the tank. What size batches have been successfully manufactured in this tank? When you're determining the level of detail to include in your initial NDA, you need to consider what it will take to make a post-approval change to an established condition in your NDA. We'll discuss change control on the next slide. Another thing, comparability protocols will be discussed by my colleague during the vaccine portion of this presentation. So 314.701I states, except for as provided for in paragraph A1II of this section, the applicant must notify the FDA about each change in each condition established in an approved application beyond the variations already provided for in the application. This notice is required to describe the change fully. Depending on the type of change, the applicant must notify FDA about the change in a supplement under paragraph B or C of this section, or by inclusion of the information in an annual report to the application under paragraph D. So what does this say? All right, I'm sorry, in 314.702, the holder of an approved application under section 505 of the Act must assess the effects of the change before distributing the drug product made with a manufacturing change. That is regardless of whether the information is contained in the NDA. You still have to assess the effects of the change before you can distribute any product. And 314.70 states that notwithstanding the requirements of paragraph B and C of this section, an applicant must make a change provided for in these paragraphs in accordance with a regulation or guidance that provides for a less burdensome notification of the change. For example, by submission of a supplement that doesn't require prior approval to distribute the product, or putting the product in the change in an annual report. So what does that really mean? That means that if you have two guidance documents and one says that this type of change is really should be a prior approval, and the same type of change in another guidance document says it's a CVE 30, change is being effected in 30 days change, then you can submit it as a CVE 30. You only need to submit notification of changes to the FDA that change a condition established in the NDA. You still need to assess the effects of any change, as we spoke about before. This is GMP. And you need to make a change in accordance with a regulation or guidance that provides for the least burdensome notification of the change. My next colleague to speak will be talking about the biologics. James is an experienced chemistry, manufacturing, and controls regulatory specialist with more than 15 years of experience and a focus in biologics and cell and gene therapies. James?
Thank you very much, Nellie. Hello, attendees. As Nellie said, my name is James Doshak, and I'm going to be talking about the appropriate level of regulatory detail for parenteral biologic applications as from a CMC perspective. Nellie, if you could advance. So first, for those of you who may not be terribly familiar with biologics, I wanted to give a quick overview. Biologic is essentially synonymous with the common terminology large molecule. They are in common parlance, grown and harvested rather than synthesized. The term is most commonly used to refer to recombinant therapeutic proteins, but it also applies to blood cells, tissues, gene therapies, and vaccines, as my colleague Rob will be discussing later. They are typically recognized as not being easily identified or characterized. They are particularly susceptible to microbial contamination, requiring aseptic manufacturing principles, and we'll go over that in some detail later on. For the FDA, at least, we recommend familiarizing yourself with the subpart of the Code of Federal Regulations covering biologics licensing and the general provisions for biologics, as these will help guide you. On the right-hand side here, I have two diagrams to kind of illustrate the change in level of complexity that we're talking about, at least from the molecular perspective. At the top of the screen, we have everyone's favorite small molecule, caffeine, which comes in at about 200 Daltons in weight, and immunoglobulin G, which is a very standard large molecule, which comes in on an average of about 150,000 Daltons by weight. So we're easily talking about 3 orders of magnitude of change. To give you a probably a more common frame of reference, this is the same, if we were talking about binary data storage, this would be the difference between a single megabyte and a gigabyte. So significant difference. But the good news is you won't need 1,000 times as many pages in your application to do this work. Nelly, if you can move on to the next slide. So I am not going to, I'm not going to itemize ICH M4Q today, as its expectations regarding biologics are pretty straightforward. But I do want to at least throw up a list of differences. These are all, every bullet on this slide is an increase in regulatory detail. Note that there is an emphasis on drug substance content. Biological applications are notoriously drug substance heavy, as formulation rarely changes following bulk manufacture. There is not a corresponding decrease in drug product detail. Don't fall into that trap. At the bottom of the slide, we have ICH, Q6B, and Q5E, covering test procedures and acceptance criteria for biotechnology and comparability for biotechnology. Each of these could be a presentation in their own right, but I will try to cover the high points as they affect Module 3. Nelly, if you would. So to start off with, I want to begin in Module 2, the quality overall summary. A few baseline pieces of advice that I would give to any client, whether they were biologic or small molecule. There shouldn't be any content in Module 2.3 that is not already in Module 3.2. We always strongly recommend that you start from Module 3 and redact to create Module 2. Don't write your Module 2 from scratch. It's a waste of time. There is a greater expectation for detail in the biologic Module 2.3 from the small molecule. You should expect it to be about twice the size of a small molecule in 2.3, so about 80 pages of text, all considered. This does exclude tables and figures, but generally speaking, your tables and figures are some of the first stuff you're going to want to cut out of your Module 3 when you're producing your Module 2. So generally speaking, it should cleave pretty closely to that 80-page mark, but it will depend on your individual product. That'll be a refrain that I repeat often in this presentation. Every large molecule is unique. Note the particular expectations regarding structure and function here for the characterization section. The agencies do not want to dig for your characterization overview, so make sure that you repeat it here. The same goes for the appendices for your facilities and equipment information. This is particularly relevant as the appendices are often passed over in Module 2, particularly in small molecule. But make sure your facility overview is copied into your Module 2 because the FDA is going to be looking for that summary information. Nelly? Moving on to general information. With regards to structure, many clients see this requirement for amino acid sequence and they ask the whole amino acid structure. And yes, the agency is looking for your entire amino acid sequence. While we're on the subject, if you're working on a cell and gene therapy product, there's an expectation for full gene sequence as well. It's pretty well understood by the industry and the agency that these guidances are not really keeping up with the times, what ought to be expected in an application, but they are still in force. So by all means, ask the agency if they want to see the whole amino acid or base pair sequence of your product, but do not make the mistake of thinking, surely they don't mean the whole sequence, and not filing it without the benefit of a meeting. It's important to remember, as I said earlier, that every large molecule product is unique in a way that small molecules are not. They are orders of magnitude more complex. While the agency is becoming more and more proficient with managing biologic submissions, it is still important to remember that we're educating them about our particular product and take that into consideration when providing detail in a regulatory submission. In the case of cell and gene therapy, this consideration is even more important because the agency is still developing their regulatory approach to these products and is still improving their proficiency in managing those submissions. In general, and again, this applies to any application, not just biologics, but is particularly important here. Please always approach the agency with respect and good faith, and never hesitate to ask for agency advice if the opportunity presents itself. It is always better to get an answer to your question that is tailored to your product if you can. Next slide, thank you so much. So drug substance manufacturing. Drug substance manufacturing is where the largest number of increased expectations for biologics can be found. Most of them are fairly self-explanatory and relate to the fact that the manufacturing process itself is more complex. The most difficult thing to remember here is to not let the increased expectations lure you into making rookie mistakes like filing too much equipment detail. Many clients propose to simply file their master batch record, particularly biologic clients and gene therapy clients. This will get you past the reviewer, but it also dramatically increases your post-approval compliance load, as Nelly addressed earlier. So be circumspect about that choice. It's fast, but it can put you at significant risk. Note here the extended focus on control of materials in 32S23 as it relates to cell banking. Cell banking is akin to a second manufacturing process, and additionally a third if you are also dealing with a viral bank. In control and critical steps and intermediates, it's particularly important to demonstrate control over your biological manufacturing process hold times and temperatures. Obviously, you pray this doesn't happen, but pronounced degradation can begin at harvest. I have seen this realization occur post-approval, and it is ugly. You really want to be on top of this before filing, if at all possible. Remember that the FDA does permit different release and stability specifications for biologics, but you should not consider this to be carte blanche. Consider the expectations of ICH Q5C and Q6B as they apply to biologics, but remember that the ideal is still a robust product that meets its release specifications throughout its retest period. Let's see, process validation and evaluation. Note that this is required for the authorization of biologics. I have heard of biologics being successfully filed in their BLAs with validation partially completed and a commitment to a plan, to an ongoing plan, but I've never been part of a successful attempt, and I would not recommend the effort. Finishing off with process development. The key element here for a biologic development module is that it should be organized with respect to risk. The agency is most interested in the cost-benefit analysis with regard to quality losses and quality gains for your product. Don't scrimp. Make sure that your justifications here are robust. You'll have a lot more text here than you would in a small molecule application. Nelly? Characterization. What we have here is a diagram illustrating the 4 orders of protein structure. On the left, you have primary, which is your amino acid chain. Secondary is the initial folding of that amino acid chain into simple structures like the alpha helix or the beta sheet. The tertiary structure is additional folding of those of the secondary structure into a protein subunit. And not every protein will have a quaternary structure. Some, like hemoglobin is a great example because it consists of subunits. If you've got multiple protein subunits that fold together into a single protein molecule, you'll have a quaternary structure as well. And all of this needs to be summarized in your elucidation of structure and other characteristics module. Obviously, this is a significant increase in detail over small molecules. Also, please remember, as I said earlier, this should be summarized in your module 2 as well. The agency doesn't want to dig for this. I don't really think biologics CMCRA folks need to be reminded about post-translational protein modifications anymore, but it certainly wasn't that long ago that we did. For those of you who might not be familiar, you know, the protein is the protein, the amino acid sequence the protein is made in the cell, but then once it's made, the cell sticks all kinds of things to it, mostly sugars, but a lot of other molecules as well. And these are important. I had the pleasure of watching the industry catch up to this in real time. R&D at a particular client, research and development, fought tooth and nail to keep post-translational modifications out of their dossier because they were convinced they'd never be able to adequately control them. Then another company a few years later started trying to develop biosimilar of their product, and suddenly their glycosylation profile was sacrosanct and inviolate intellectual property. The lesson here is that, yeah, it can be difficult to control these, but even when they're inconsistent, understanding and being able to describe those inconsistencies is still important. We're still learning about how post-translational modifications impact product quality, and what seem like footnotes today could be headlines tomorrow. Next slide. I'm finishing off with the discussion of the appendices and regional information in 3.2a and 3.2r. The facilities and equipment appendix is a major focus for biologic applications. I once had an R&D manager tell a regulatory affairs colleague that he could manufacture their biologic in the parking lot on mobile skids, as long as he was honest with the agency about it. I like his faith in the regulatory compliance process, but I don't think he'd have a lot of success. As I mentioned earlier, these products cannot be sterilized in the simple fashion of a small molecule. So facility cleaning, sterilization, flow control become extremely important. Maintenance of those aseptic procedures is critical. Documentation of these processes and adherence to these documents upon inspection is absolutely paramount. For the FDA specifically, we recommend familiarizing yourself with Code of Federal Regulations, the subpart regarding establishment standards. It'll guide you to a large extent. And also remember that this facility, the summary of this facility information, don't copy it all into Module 2, but the summary of this facility information should be in Module 2, because again, the agency wants to see that right up front and center. They want to make sure that you are on point with this. Adventitious agent safety evaluation could be its own presentation, and regrettably, because I am already coming up on time, I think I'm going to gloss over this. It is a very broad topic. It is critically important, making sure, because the thing about a biologic is that, you know, any therapeutic product can develop issues with contamination, but only a biologic can only in biologics can your manufacturing process actually get sick and possibly die, or worse, hurt your customers, your patients. So we recommend that you review ICH Q5A, Q5D, and Q6B. Focus on the selection, testing, and safety assessment of your cell and viral banks. That's really, you know, where you have the most delicate issues. But also remember that for both facilities and equipment and adventitious agents, you've already touched on many of these topics elsewhere in Module 3. So leverage your cross-referencing to minimize redundancy. But at the same time, don't make the reviewer work to understand your key messaging. You can restate your control summaries here in your drug substance and drug product modules, and also in Module 2. It is a balancing act, but that's generally a good thing. Restate your summaries wherever you can, but make sure that when you are including large chunks of data that you cross-reference rather than repeating. That's it for that slide, Nellie, if you would. So I'll be handing off at this point to Rob Waterland. Rob is a valued colleague and an experienced technical and medical writer whose expertise is in biologics and cell and gene therapy, like myself. He's also a resident expert on vaccines, which is what he's going to be addressing today. Rob, you can take it from here.
Okay, thank you, James. So as advertised, I'm going to talk about CMC aspects of vaccine filings. Next slide, please, Nellie. So throughout this talk, I'm going to highlight a small number of key takeaway messages. And the first one is on this slide. In the US, vaccines are regulated by FDA as biologic drugs. So everything that James has just said applies for vaccines. And indeed, it is the case that if you look at a vaccine IND or BLA, they're typically very similar to the ones that you would have for a typical biologic product, like a monoclonal antibody. So that gives me some liberty to speak about things other than the details of the biologic application, because James has already covered it. So what I'm going to talk about mostly is the history of vaccine approvals, the steps that have been taken to accelerate vaccine approvals, and the need for future regulations that have come up as a result of the response to the COVID crisis. So you can see the pathway here for the approval of a vaccine. It's the standard BLA pathway. An IND is filed, clinical trials begin. If everything goes well, a BLA is filed. There's a pre-approval inspection at all the manufacturing sites. And finally, there is an FDA advisory committee meeting to judge whether the BLA will be approved or not. I've included at the bottom of this slide the current FDA guidance. I don't expect you to memorize this, but you'll know where to find it. It's there mostly to show you that the guidance, existing guidance for FDA for vaccines is quite old. So next slide, please, Nellie. So what makes vaccines distinct? Vaccines historically have had very long development lifetimes. So in 1996, it was estimated that it was about 10 years from preclinical to launch. And 2010, it was estimated that it was 15 years. So why is that all true? It comes from a variety of causes. A group of industry and academic thought leaders met and examined what the barriers were to vaccine development and approval. And they came up with a list, and the three most important ones are listed in this table. Two of them are financial. They are reflecting return on investment. So the pharmaceutical industry has the view that there's limited return on investment for vaccines that address diseases that very few people get. And in addition, there's a limited return on investment for vaccines compared to non-vaccine projects. That's an opportunity cost kind of argument, if you like. Academia, on the other hand, noted that it had become very difficult to progress advanced vaccines beyond proof of principle. So in fact, if you go and look at the list of vaccines currently licensed by FDA, you'll be a bit shocked to see that it's surprisingly short. In the last 10 years, there have been 14 vaccine BLAs approved, but 5 of them are for diseases that would be precisely in that limited market size rubric that manufacturers don't like. Next slide, please. So what did FDA do about it? This is not just a problem for vaccines, it's a problem for drugs in general. So FDA has, of course, instituted regulations to accelerate drug approval for serious conditions. There are 3 main programs. Accelerated approval, which gives permission to market a medical product based on a surrogate measure. That's really important for vaccines. That surrogate measure is usually antibody levels for vaccines. There are 2 programs, Fast Track and Breakthrough Therapy Designation, that where FDA prioritizes certain programs during clinical development, and most importantly, permits rolling review of the filing. In addition, there are 2 programs that authorize special access to medicinal products before FDA approves them. There's the There's, of course, compassionate use or expanded access. And then most famously now we have all become used to emergency use authorization, or EUA. The word authorization is underlined because FDA is very clear on this. There is no approval associated with EUA. You are merely authorized to use the drug. The expectation is that a BLA is filed. So how does EUA work? There's a presidential declaration of a public health emergency, and after that, FDA can allow use of a product, including a vaccine, before it has been demonstrated that they meet the usual FDA approval criteria. So this came in, emergency use authorization was available to FDA after 2004, extremely sparingly used until the COVID pandemic. So next slide, please. So did any of this work? This slide sort of shows that indeed approvals have accelerated. The timelines, the boxes in blue and yellow with the dates on them indicate the points at which FDA promulgated these new regulations. And the red circles with numbers inside them are the number of vaccines that were approved in the interim. So between 1987 and 1992, there was only one vaccine BLA that was approved. More recently, approvals have accelerated, and there are now approximately one per year. Is this causation or correlation? Who knows? Did the regulation stimulate this? Not important. Vaccine approvals have accelerated. I will point out that many recent vaccines were improvements on existing vaccines. So very recently approved is the Merck Gardasil 9 vaccine for diseases caused by the human papillomavirus, HPV. That stands alongside the previously approved Gardasil, and it adds an additional 5 additional HPV types. So to some degree, there's a little bit of like a Me Too-ism, if you like, in vaccines. So next slide, please. Let's talk about vaccines themselves. A lot of talk going on about what's in them, how do they work. There are a small number of strategies for designing vaccines. Generally speaking, vaccines fall into 3 classes. There are first generation, second generation, and third generation vaccines. And the first generation and second generation vaccines are the ones that I think pretty much everyone on this call received as when they were a child. They consist of first generation vaccines are whole virus, either weakened or inactivated. Second generation are subunit vaccines, where parts of the virus or bacterium or a bacterial toxin is what the active ingredient is in the vaccine. I will point out, it's easy to forget this, can you go back, that you know, we do have vaccines for bacterial diseases. You think of them for viral diseases, but if you think of the DPT vaccine, diphtheria, pertussis, and tetanus, that's a vaccine for bacterial diseases only. So it's good to remember that. So next slide, please. So third generation vaccines, they've been in the news. They don't provide an antigen directly. What they do is they provide a genetic code for your body to make part of the virus. And currently we have 3 flavors of them. We have the viral vector vaccines. An example of that would be the Johnson & Johnson vaccine or the AstraZeneca vaccine, which doesn't have emergency use authorization in the US. And there are messenger RNA or DNA vaccines. So the viral vector vaccines, what you do is you provide DNA for a specific viral protein, insert it into a different harmless replication incompetent virus, typically an adenovirus, and that virus is injected into the patient, into the recipient, and enters the cells, and the viral protein is expressed. Now, for 30 years, it's been known that if you inject naked plasmid DNA, bacterial plasmid DNA, or naked messenger RNA into a muscle, the muscle cells will actually express the protein that the DNA or messenger RNA codes for. So that's the basis for messenger RNA and DNA vaccines. And of course, they've been very much in the news. So let's go on to the next slide, please. I'm not going to dwell on this. These are lots of vaccines listed here. Let's just focus on the text that's shown in red, which is for the messenger RNA and DNA vaccine. So you can see there are 3 listed there. The first 2 are messenger RNA vaccines. They are the Moderna and Pfizer COVID-19 vaccine. And also listed is ZyCoV-D, which is the very first in history human DNA vaccine that has been given emergency use authorization. Last week, the Drug Controller General of India approved it for emergency use authorization. So the reason I'm showing this slide is that you can see there the COVID pandemic and the response to it has generated an enormous change in the range of technologies that are available for designing vaccines. So next slide, please. So we got to talk about messenger RNA vaccines. Is it the dawn of a new age? There's certainly a lot behind them. Why are they exciting? They're exciting for a number of reasons. First of all, the vaccine design is trivial. Once you have the target, the sequence for the target, let's say in COVID, it was the Chinese publishing of the DNA sequence for the target. It was very easy to design. In fact, it took 2 or 3 hours. So if you wanted to design a COVID vaccine for a new variant, you can design it very, very quickly. Most importantly, they are extremely easy to manufacture at commercial scale. And the primary reason is because they do not use human or animal cells. There's no eukaryotic cell expansion and transfection. There's no cell banking. There is, in addition, no viral banking. So these are just manufactured using bacteria, using standard biochemical processing. Bacteria reproduce really fast, so very quickly you can produce enough vaccine for animal testing. Because there's no cell banking, because there's no cells, because things are clean, the purification of the vaccine is very straightforward. So they're very easy to manufacture. Despite that, we know from COVID that they're very non-infectious and they stimulate very strong immune response. What I think is less well known is that they have lots of potential uses. So of course, there are active vaccine pipelines on the way to produce messenger RNA vaccines for things like Ebola, rabies, Zika, tuberculosis, HIV, influenza, you name it, you'd expect that. But there are also true cancer vaccines under development. So you design a messenger RNA vaccine to specifically target proteins that are expressed, that are made by cancer cells. In June 2021, the first dosed patient was dosed for a vaccine for advanced melanoma, phase 2. So they're targeting what are usually called cancer markers in the oncology community. Better yet, you can do straightforward gene therapy with messenger RNA vaccines. Cystic fibrosis is a disease caused by mutated proteins. You can use messenger RNA vaccines to provide the correct proteins. So this is all very exciting. And it's certainly true to say that messenger RNA represents the most important development in vaccines in decades. So that's great. What are we going to do about it? Next slide, please. So unfortunately, because the global response to COVID came so fast and catalyzed this cascade of novel vaccines, FDA's guidance just doesn't reflect that. I mentioned before that FDA's guidance is pretty old and reflects the status quo of a few years ago. In general, a new technology will require new regulatory and oversight mechanisms. As I've mentioned, messenger RNA vaccines, DNA vaccines use genetic material, and they are to some degree genetically based therapies. In fact, you can argue, are they vaccines or are they gene therapies or are they both? Whatever the case is, it's clear that FDA is behind the curve on providing guidance and maybe regulations on this really important vaccine development. At a minimum, a messenger RNA vaccine specific guidance is urgently needed. So now I'm going to switch gears completely and go to nuts and bolts. Next slide, please. Nellie mentioned this a couple of times. I'm going to talk about the use of comparability protocols in NDAs and BLAs. So this is not vaccine specific, it applies to anything. So the FDA encourages the use of comparability protocols and actually feels like they're badly underused. What are they used for? They're used to manage post-approval changes. What are they? So comparability protocols, or CPs as they're called. The CP is a document. It's a prospectively written plan that's filed in 3.2R that shows how the effect of a proposed future CMC change will affect the quality and potency of the product. So it's basically a roadmap for doing the comparability of the pre and post-change product. So why are they useful? For the agency can pre-review them. They can also pre-review the plan that's contained in them to implement the change. And from the point of view of the manufacturing authorization holder, what's great about them is you can propose a reduced reporting category for the change. So for example, suppose you expect that in the future you'll be bringing into make additions of additional drug product plans to your manufacturing network. You can file a generic DP site comparability protocol and file that with your original BLA. The agency will review it and hopefully approve it. And if they approve it, instead of having to file a PAS, a prior approval supplement, which of course takes a long time, you can say, and the agency can agree, that you can file it as a CBE 30. That's a fantastic thing. So be mindful that these are useful and should be used more. So my last slide is on drug master files. And drug master files must be the most dull topic in all of CMC. But they have a use that I think is not terribly well known. So drug master files are typically used when there are two manufacturing partners for a drug product. And the DMF filing process allows each party to protect its intellectual property, allows the agency to review efficiently. But the DMF process can also be used to simplify dossier maintenance. And this is particularly true when you have a manufacturer who is using a contract manufacturing organization, a CMO. So the DMF process can be used to provide confidential facility information for the CMO within the marketing authorization holder's filing. And it will basically get filed in 3.2A1. I mean, there'll be a minor filing in 3.2A1 with very summary information on the CMO's manufacturing facility and something that says all the details are contained in the DMF. And here's a reference to where you can look for it. So how does that help? So if the CMO makes some post-approval change, they update their DMF, they provide new letters of authorization to FDA and their partner manufacturer. And the partner manufacturer, the DMF, the market authorization holder, doesn't have to make any significant change at all in their approved dossier. So manufacturers do this, and they probably should do it more often. So I think that's my last slide. And now I think we'll go into the question and answer session.
Excellent. Okay. So let's go through here. I think we have a couple that are voted up here towards the top. This first question asks, can you comment on similarities or differences in drug products for 3.2.P requirements for mRNA vaccines and lipid nanoparticle formulations versus small molecules in liposome formulations? There's an existing FDA guidance on liposome drug products.
Yeah, good question. I can't specifically comment on the details because we don't have a new guidance. I think they're similar. The level of detail for the messenger RNA filing. I'm sure FDA is going to continue to regulate them as biologics. So they will have a heightened level of detail, as James pointed out. But I think the underlying question would be, would the agency accept, because the manufacturing process is so much easier, much less detail? I suspect not. I suspect not.
Excellent. Okay, our next question asks, generally, how should a BLA be tailored? For example, amount of detail, other considerations for a cellular therapy product.
Ooh, that's not a small question. I mean, in general, I mean, it should be tailored the same as following the biologic and cell therapy guidances that the agency has put forth. That's very broad. I'm not really sure how to go into more detail on that at this point. I'd be happy to answer more particular questions via email.
This other question, let's see, I'll move over to the chat here just to address some of the ones that were entered here. This one asks, are there any interaction between an inactivated and mRNA vaccine? And they continue on to say, like as we take the flu shot and booster, say Pfizer, what duration or interval should be between the two?
So another great question. We don't know. My understanding is that there are some early studies looking at possible effects of that type. There is also talk, at least in Europe, of having a combined messenger RNA booster and flu vaccine. So I think it's a case of watch this space. We don't know yet.
Our next question asks, this is back in the Q&A, says this may be more of a legal off-topic question, but regarding biosimilars, do the authorities compare the originator and the biosimilar applicant documentation for inconsistencies regarding characterization, for example, if the sequences are actually the same?
So I can't speak to what the agency does behind closed doors, but what I can say is that characterization is a lesser concern. They're primarily concerned when it comes to biologics with safety and with efficacy, with purity, potency. So at least where the sequences are concerned, I think that would only be a secondary concern at the most. Generally speaking, it's more a question of, you know, does it do what's on the tin? Is what it does similar to the reference product? And again, like, is it safe? Is safety represented? So I think that the broad answer is no, but there's probably more nuance to that I can't really speak to.
Alrighty. Let's see. The next question we have is regarding S23. I was recommended by a colleague to include only pictures of the chosen clone for monoclonality prior to MCB and WCB. What do you think about that?
I've never tried it. I wouldn't recommend it. I think standard wisdom when it comes to regulatory is that you want at least one corroborating set of data when you're talking about identity. Obviously, it's a little bit different because you're talking about your cell line rather than your actual product. But in general, I would say back yourself up with something. I wouldn't just go in with a picture and call it a day.
Let's see. Our next question asks for a vaccine BLA. It says QoS 2.3 for a product containing more than one drug substance. Approximately 80 pages apply for each drug substance and drug product. So asking in total around 80 pages, 80 times 3 pages. I might need some clarification there from.
I think this one's intended for
slide, right?
No, I mean, it says vaccine, but I think that, I mean, I certainly hope not. I mean, it's possible. I will say that. I think this really comes down to your product and what is required. You know, how if you have 3 completely independent drug substances, which is my understanding is that's typically how vaccine BLAs work, Rob.
Right.
If you want to comment on that, yeah, it is entirely possible.
Yes. Indeed, if I can comment on that, I mean, so there are now vaccines that seem to put almost every childhood disease in the same combination, right? You have DPT, chickenpox, HIV, you name it. It doesn't scale linearly with the number of drug substances. You know, if you have 3, it wouldn't be 3 times bigger.
Mm-hmm. You may have to do a lot of cross-referencing. I would think if you've got if you have like the same manufacturing facility for 2 of them, you could always cross-reference that information. In general, cross-reference whenever possible. It is a great tool. It is the benefit of the modern hypertext age that we live in.
Let's see. So we are at 12 o'clock. While I'm reading this next question, I'll just put the completion code on the screen for everyone. We'll address one more question here for everyone. So there's the completion code on screen. And then our last question here that we'll address just asks, where can we find guidance regarding comparability protocols? For example, manufacturing scale up and proper statistical analysis to follow.
So there is direct guidance available on the FDA site. If you just search for comparability protocols, you'll find it. If you're interested in filing also in the EU has basically an identical document, which is called a post-approval change management protocol. You can look for EU guidance on that. And comparability protocols are also discussed in ICH documentation. So there's lots of places to look.
So I've just
also,
oh, sorry.
I was just going to say, also ask questions. You know, again, like comparability protocols tend to be very specific. So reach out to the agency, ask questions.
Yes, Nellie had a slide very early on in her talk that said, get to know your regulatory program manager.
Yeah.
This is an exact place where you should talk to them. The FDA is encouraging the use of these, and they can help guide you.